stage 1 on the upper left, stage 2 for the breast cancer, and stage 3 when you can see that the little bits of cancer have broken off. And then stage 4 is when it's actually dislodged into the lungs. You can see that. So the cancer from here has gotten into the lungs. And summarizing this is that we have 4 stages of description. Stage 1 is basically 0/1 is when it's limited, very small. Stage 2 is when it's become invasive and actually grown to the original tissue around the breast tissue, and possibly, not necessarily, but possibly broken off. It's hard to trace that, right? It's really hard to track if any cells have broken off. Stage 3 is when you have it broken off and now it's beginning to have the ability to spread. And stage 4 is when the cancer has completely broken off to another organ system. And that's where it gets really scary. All right? And so what I want you to think about now is which of those stages, which one of those 4 stages do you think is best used to determine the stage of cancer? All right? So when you think about all of those different stages and what I just talked about, which one of these or all of them do you think is the best way to determine which stage of cancer it's in? So number 1, the size of the tumor. B, the presence of the cancer nearby the lymph nodes. C, whether the cancer has moved to another organ system of the body, or if all of these are pretty legit considerations, right? So when you're a doctor, when you're a physician, and you go see a patient, and then they come out with a stage 0, a stage 1, stage 2 cancer, and a doctor needs to be able to make the best decision, what kind of characteristics is he or she considering, right? And again, this is not going to be graded on accuracy. Just shoot your best choice and you get the points. All right, so just make sure to put in your answer. We have another minute here and we're going to keep the ball rolling. So make sure you're logged into TopHat. So during the Zoom time, if you are on a laptop or if you are at home, make sure you have a device that is connected to TopHat so that way you can just follow along. All right, so I'll give you guys another 45 seconds, folks, to post an answer. And make sure to read all the choices, right? The last choice here is all of the above are correct, right? So make sure to read every choice. Make sure to read the question thoroughly before you select an answer. All right, these are good practices to taking multiple choice exams, especially in the field of biology. There's a lot of content, a lot of information, and a lot of the ways that assessments are presented are through multiple choice. All right, T minus 9 seconds here, so post an answer. All right, here we go. Here we go. All right, so let's take a look. All right, so the answer is all of these are correct. All right, great job. So 93% got it right, nailed it. Okay, so let's go back to how this affects our body. So our body normally has ability to maintain balance. And why is cancer so nasty and so detrimental? Well, homeostasis is your body's ability to maintain health and be able to balance out not only your blood pH, your temperature, your glucose amount, the sugars, the ions, the salts, the amount of fluid your body has, but it maintains a natural set point, which is what we consider as like a range that's healthy. Right? So the word I want to throw out into today's discussion is called set point. And set point, if you think about, is none other than an imaginary value that allows you to maintain health. All right, so set point, let's write this out here. So the word set point, this means it's an ideal range that your body wants to be at. So for example, the pH of your blood is ideally about 7.4. But let's say you woke up this morning and you drank like 2 cups of espresso and you had some more caffeine. All of that caffeine is probably going to affect the amount of water in your body, causing you to change your pH a little bit, right? Maybe you ate a lot of protein in the morning, and now the protein intake in your diet is also going to affect your pH. And now your pH is a little bit lower, so your body's more acidic. So there's these fluctuations that happen naturally. We can't control every little detail of our lives, but you want it to be in a healthy range. When you have cancer, cancer will affect an organ, which will then affect the ability to maintain these balances, right? So you have your liver on the far left that plays a role to detoxify your blood. You have your heart that maintains the blood flow and the blood pressure. You have your respiratory system that gets oxygen into your blood cells and getting rid of CO2. And then we have your kidneys that help you dilute and get rid of any waste, process penicillin, process toxins in your body, excrete waste out. So when you have one of these systems affected, just imagine you had a cancer in your lungs. Now you can't do the oxygen and CO2 exchange properly, which is going to affect your blood, which is going to then, the blood goes into your other tissues, which will then affect other cells and other systems, right? So there's this consequence and this domino effect that occurs when one system gets damaged or isn't functioning to its maximal capacity. And tumors, oftentimes, when it has a cancer in the body, it takes up a lot of space. It grows. If it happens to be in the wrong spot, it could block the function of blood flow. It could block the ability for cells to exchange gases and therefore deprive cells and starve them down. And that's what's really nasty, is that it's so hard to point out any cell, because any cell that has the ability to divide potentially could be cancerous. And can we possibly live in a vacuum? Probably not. Right? So that's really the hard facts here. So which of the following terms means the body's ability to maintain and operate under set defined internal condition, right? So a set point. So this is an easy one. So just go and quickly answer this one. Which one of these terms represents that balance that I just talked about? Your body wants to be at, but can you always maintain it? Not necessarily. Yeah, Mo. At the end, after the class ends, I can reopen the questions. All right. So we'll just keep on going, and then if you miss a question after the class ends, I will reopen them. Just stick around if you miss the question. All right. So go ahead and answer as soon as you can. We have another minute. So this is why it is important that you are following along on top of that, because I can't spend more than 2 minutes on each question, because that will consume too much of our content time. And so just please try your best to follow along, key and answer, right? You don't have to get it right. Just post something in there. Try your best to recall what you heard me talk about. All right, so another minute here. And we're going to keep on pushing through. I don't think I might be able to cover all the slides today, so that means next Tuesday when we start our session, even though we're going to begin a new module, I'm going to wrap up the last few slides if I don't cover everything, all right? Because I have a feeling that I've spent a little bit more time on our discussion than I should have, but it's all good. All right, so another 30 seconds, people, so key in your answers. Make sure your phones are charged. Make sure your iPads are charged, right? You don't want to start our Zoom session in the morning and your battery is like low battery sign pops up. So that's always not a good feeling. Start the morning off with low battery, right? That's not a good sign. All right, T minus 9 seconds here. All right, this one should be easy, right? 4 seconds, here we go. 3, 2, and 1. All right, so let's take a look. So the answer is awesome. Most of you guys said D. Answer is homeostasis. Great job, guys. Good job. All right, so now how is cancer treated? So in order to treat cancer, we have to be able to diagnose and find cancer. And this is where things get exciting. So one way that we could find out if you have cancer is also parallel with what you've been hearing about literally almost for the past few years, PCR testing. Right? You probably heard this more than you should have because of the COVID and trying to track if a patient or somebody has contracted and tested positive. But PCR is a device, literally a machine, a device, that will take a little bit of DNA and amplify it into numerous segments so that it becomes visible. So when you hear the word PCR or polychain reaction, it's a machine that will take a little sample of DNA and with the right amount of reagents, break it down, heat it up, and anneal it so that it keeps on growing and growing and growing, eventually making a lot of that DNA from that one small little strand. What I want to go over is what PCR really is. So when you hear the word PCR, you're probably like, what the heck is that? How does that work? Well, basically it's a machine, and the machine has a certain set temperature where you take a little bit of DNA and you put it into a small little vial. And even though this is a cartoon image, you can see that it's a machine where you basically place the DNA into these little wells, and oftentimes there's about 96 wells or even more, depending on the size of the machine. You close it down and you literally let it heat up and begin to break down. So what this machine does is that it breaks down the DNA and it splits the strands from the DNA that you see in my background here, the 2 strands. It splits it in 2 strands and begins to allow other different kinds of primers, which are added pieces of chunks of DNA, to grow that strand and make it bigger and bigger. So you start off with 1 strand, right? The science behind this is really fascinating. Is you start off with 1 little piece of DNA over here, you put it in the machine, and it heats it up and it breaks it to 2 little strands. Now you would add other reagents in there, including primers and different enzymes, and now that will take those separated strands and begin to grow that 1 strand to 2 strand. And then it will then do that again, break those 2 strands down, and then grow it into 4 strands. So the whole point of PCR is you have 1 little bit of DNA, and by the end of whatever cycle you set it to, you get a tons of that DNA. And when you get a ton of DNA, you can actually see it with your eyes. You can actually see it and visualize it with a gel. And the tricky part about doing this whole process is that you need to find the right temperature, because DNA is very delicate, right? It's a very temperature sensitive. And if you heat it up too much, it will just literally decay and completely get damaged and destroyed. And then if you get too cold, it's not going to do anything. So that's really the science behind it. It's a machine. And after you do all of that stuff, you can take the final product of your DNA that's been amplified and then put it through computer sequencing and tag it up and look at what exact bases do you have. And when you look at this screen, you're like, I don't know what I'm looking at, Lynn. This is kind of confusing. So what you're looking at is after you get that final product, you would put it into this gigantic computer, and the computer has the ability to tag the different nucleotides of the DNA with a laser and literally read which kind of bases is found at which area. So over here on the bottom is an example of what you would see. The computer would then tag, oh, the first base is G, and it's this peak because that shows you it's that base, T, C, and then these waves and peaks represent the kind of nucleotides and also the number. It literally will just give you this gigantic long paper of bases and waves. And then when you read it, you can literally code, okay, well, if we start over here, we have the first 3 bases. GTC is the first set of codes. And then the next set of codes is ATT. And next one is ACA. And that's literally how they decoded the DNA and the presence of COVID viruses in human samples. So you take a human sample, right, from your saliva or your tissue or your blood, and then you can sequence it and see if you see that sequence inside your blood. If you see it in there, that means it has to be COVID, because normally we don't have that sequence in our body, right? So it's a way to guarantee that something's in there. And then, of course, with cancer, if you know what kind of cancer mutation you're looking for, you just screen for that as well. So it's a very time-consuming process, and that is why when there are cancers that are more widespread and more common, they tend to have a lot more focus on them. As a result of all this breakthrough and these kind of technology, you can then develop something called a DNA microarray or gene chips. And these are really cool. Gene chips are little pieces of segments of DNA readers that are already set to look for specific mutations. So for example, let's say they've studied like 10,000 breast cancer patients, and they realize that almost in 99% of all these breast cancer patients, they had that same mutation. So what they then do is they take that same mutation and they just look for that one mutation, because the chances are pretty high, right? Instead of looking for that 1%, you're looking for that 99% mutation. And most likely, if you have a mutation, they can probably, with pretty good confidence, say, hey, maybe there is something wrong in your cell. Maybe there's a little bit of cancer that we can start thinking about how to fight it, how to treat it. And of course, there's a lot of other protocols involved as well, using antibodies to test for it, but the DNA way and the way to use PCRs and DNA chips and microarray are really powerful because they're very accurate. And oftentimes you can also do complementary pairing with mRNA, but that gets a little bit more complex, so we're not going to jump into the details of that. But just know that these chips are very specific because we know that there are certain mutations that are highly shown in numerous cases of the same kind of cancer. So another way to do this is utilizing a blood test. And this is pretty cool. Some of you probably have done it before. I'm actually getting one done not too late in the semester. I actually have one set up. I do an annual blood test, and I can even share it if you guys want to see what it looks like. But CBC, not CDC, but CBC is what they call a complete blood count test. And they will look for the basic chemistry, the basic morphology, and the shape of your blood cells. That's your red blood cells, your white blood cells, your platelets. There are a different set of white blood cells in your body. You might have known this, you might have heard this before, but there are actually 5 kinds, and there's 2 classes. The white blood cells are divided into what they call the granulocytes, which are these 3. And then we also have what they call the agranulocytes, which are these 2. All right? You don't need to know all those details yet, but just know that when somebody says to you white blood cells, it's kind of vague. There's actually 5 classes. And then the red blood cell, which is these little human red blood cells, RBCs, are pretty classic. Right? They look like a little donut, and they don't have a nucleus because their job is simply just to carry oxygen. But their function really is based on shape, because if they're healthy and their shape is right, then that means they're performing their job. White blood cells, though, on the other side, you can see they have these weird, irregular-shaped nucleuses, and I'll just highlight them with a little marker here. You can see that some of these have this multilobular nucleus, which looks completely alien-like. You also have an eosinophil, which has 2 lobes, and then you have this lymphocyte that has 1 big lobe, right? So you can see that the shape and then the structure of these cells are kind of different. And that's why when you do a complete blood count, usually a well-trained scientist or technician will visualize your cells and just kind of get a quick glance, look at any kind of potential morphology issues, and of course looking at if there is an abnormal number of certain kinds of cells. So there are certain kinds of proteins that can be screened in the blood, and there are 2 that I want to just post out there. There's one called the CA153 and one called the CA125. These numbers might not make too much sense to you, but it is good to know that these proteins are specific genes and that are coded by DNA that get expressed. So whenever there are mutations or blood cancers, oftentimes you might see these proteins in your blood, right? Which shouldn't be there. And so if you don't see them in there, that means, okay, your cells are probably doing good. Right? So that's why doing a blood test, I would say probably annually or at least once every few years, is pretty good in terms of preventative medicine, right? You want to know what's going on in your system. And when you see changes to the way your blood behaves, changes to the ion level, changes to enzymes, that could be an indicator of some underlying issues. All right, so I am getting close to the end of the class here. I want to share one more slide before we wrap it up for today. There are a lot of different kinds of ways to scan for cancers, and just like you guys talked about today in the discussion question, right, there's a lot of ways that we can use really nice, expensive, high-tech devices to visualize cancer. Some of these ones include an X-ray, right? So using an X-ray to look for any irregular tissue, for example, X-ray for the breast tissues. You can also use a mammogram, which is a very specific way of looking for a breast tissue cancer and also having a doctor assess for lumps. You can use what they call a CT scan, a computerized tomography scan, which takes a 3D generated version of an X-ray to look at any kind of irregular distortion. Another one that's pretty dope is called the PET scan, which stands for the positron emission tomography. These ones are a little bit more invasive because you have to actually inject a dye into the body, and that dye gets uptaked into your cells. And based on your cells' ability to respond to dye, it gets contrasted out. So that's why there's some folks that might be very, very hyper responsive or sensitive to these dyes, and that might cause discomfort, and the dye feels like it's coursing through your body and into your veins. And the last one is what they call MRI. Right? MRI is when they use radio waves that blast it through your body and they generate a 3D image.